DNA or deoxyribonucleic acid are double helix strand that holds the gene that affects the traits of our body. The DNA molecule is composed of phosphate group, sugar deoxyribose, and 4 nitrogenous bases, which are adenine or A, thymine or T, cytosine or C, and lastly guanine or G. A or adenine will always be paired with thymine only in DNA, and thymine will be replaced by uracil if it is in RNA or ribonucleic acid, while guanine is always being paired with cytosine even in RNA. Deoxyribonucleic acid differs in RNA by strand counts, base pairs, and lastly sugar. DNA has 2 strands or double helix, while in RNA it only has 1. Thymine is on DNA and being replaced by uracil if it is in RNA. And lastly, DNA has deoxyribose sugar while RNA have ribose sugar. Central dogma is a process in which the DNA replicates itself to make RNA and RNA transcripts and translates its bases to make proteins. Inside the cell, we have nucleus where we can find all of the chromosomes and start the DNA duplication. In duplication, an enzyme called helicase breaks the bond between the bases until the DNA splits. Then free nucleotides fill up the bases, and with the help of DNA polymerase, it forms new strands. And also by the help of primase, the DNA polymerase can start and stop the process by putting primer to start the polymerase. 5' to 3' is the leading strand while 3' to 5' is the lagging strand. The primase works twice because DNA polymerase can only work on 5' to 3' strand. And after that, Okazaki fragments comes in to fulfill the spaces and ligase glued down those fragments in the lagging strand. And the lagging strand splits and an RNA is formed after some adjustments on it. After the duplication of the DNA, messenger RNA is created and go outside on the nucleus to come into the ribosomes. Messenger RNAs get attached into ribosomal RNA to use its information to make proteins. Transfer RNA translates the code of messenger RNA while bringing the amino acids to make protein. Amino acids make peptide bond until stop codon deter the peptide bond, and lastly peptide bond is being created as protein by converging all of the amino acids.